Grade 10 Biology Lesson How do organisms reproduce? Module 3 Objectives. At the end of this module, you will be able to identify the parts of flower, differentiate unisexual and bisexual flowers, understand pollination, illustrate double fertilization in flowering plants, list out the post-fertilization changes, identify the parts of dicot seed. Sexual reproduction in flowering plants. The reproductive parts of angiosperms are located in the flower. The parts of the flowers are sepals, petals, stamens, and carpels. Sepals are green structures that protect the inner parts when the flower is in bud stage. Petals are colorful and attract the insects for pollination. Stamen. Stamens are male reproductive parts and produce pollen grains that contain male gametes. Each stamen has two parts, a filament that is a stalk, and anther, a solen top part which has large number of pollen grains. Carpel. Carpel is a female reproductive part and produces ovules that contain female gametes. It has three parts, stigma, which is top sticky part and receives pollen grains during pollination. Style, which is a middle long part. Ovary, which is a solen part and contains ovules. Each ovule has an egg cell that is female gamete. Unisexual and bisexual flowers. Unisexual flowers, the flowers which contain only the male or female reproductive organs are called unisexual flowers. Examples: papaya, white mulberry, and watermelon. Bisexual flowers, the flowers which contain both male and female reproductive organs are known as bisexual flowers. Example: hibiscus, mustard, and tulip. Sexual reproduction in plants steps. Following steps together complete sexual reproduction in plants. They are pollination, fertilization, seed formation, and germination. Pollination. The process of transfer of pollen grains from an anther to the stigma of the flower is known as pollination. There are two types of pollination. First, self-pollination. The transfer of pollen grains from the anther to the stigma of the same flower or another flower of the same plant is called self-pollination. Cross-pollination. The transfer of pollen grains from the anther to the stigma of another flower of a different plant of the same species is known as cross-pollination. It generally takes place with the help of some agents like insects, birds, wind, and water. Fertilization. Pollination is followed by fertilization in plants. Fertilization is the process of fusion of male and female gametes to form a zygote during sexual reproduction. Events of fertilization. Pollen grains land on the stigma of the ovary. Pollen tubes grow out of the pollen grains, travel through the style and reach the ovary through micropyle. Pollen tube has two male germ cells. Each ovule has two polar nuclei and a female germ cell or egg. Pollen tube releases two male germ cells inside the ovule. One male nucleus or gamete approaches the egg and fuses with it to form a diploid zygote. This is the first fertilization and this fusion is known as synchrony. The other male germ cell fuses with the two polar nuclei and form primary endosperm nucleus or PEN. This process is known as triple fusion. So, in flowering plants, two fusions take place during fertilization. Thus, double fertilization occurs in embryo sac, which is unique in flowering plants. Post-fertilization events in a flowering plant. Events which take place in a flowering plant after double fertilization are termed as post-fertilization events. Important post-fertilization events are development of endosperm from primary endosperm cell, development of embryo from zygote, and development of seed from ovule, last development of fruit from ovary. After fertilization, the following changes takes place in the flower. Zygote divides several times and forms an embryo inside the ovule. The ovule develops a tough coat and changes into the seed. The ovary grows rapidly and ripens to form a fruit. Petals, sepals, stamens, style, and stigma shrivel and fall off. Endosperm. Primary endosperm cell formed as a result of triple fusion undergoes repeated divisions and a triploid endosperm tissue is formed. The main purpose of endosperm is to provide nutrition to the developing embryo. Embryo. Embryo is a diploid cell developing from zygote. Development of zygote to form embryo starts only after endosperm formation has started. This is because endosperm provides nutrition needed for the embryo to develop. Seed. Seed is a fertilized ovule. Basic structure of a seed consists of the following parts. Seed coat. Outer covering of the seed, which is composed of two layers, testa, thick outer layer, tegmen, thin inner layer. Radicle. It is the embryonic root which later gives rise to the entire root system. Plumule, known as embryonic shoot, which later gives rise to the entire shoot system. Cotyledons, known as seed leaves, function food storage, provide nourishment to the developing radicle and plumule. Hilum, a scar on the seed coat through which seed attached to the fruit. Micropyle, a small pore on the seed through which oxygen and water exchange occurs during seed germination. This is a structure of dicotyledonous seed. This image shows the structure of monocot seed.